The last kind of research that we're going to talk about is experimental research. This is what most people think about when you talk about scientific research, experiments. So let's talk about experiments. Experimental research is going to be different from both descriptional and correlational research because in this case we're going to involve manipulating a variable. We're not just measuring variables in an experiment, we're actually manipulating at least one of the variables, and we're still going to be doing some measuring, but we're going to be trying to measure the changes in another variable that have been caused by that manipulation. And the third thing that we need to keep in mind, in addition to manipulating one variable, measuring another, is that we're doing this all while holding other factors constant. We're trying to control for other variables. This is why most experiments are run in a lab, a controlled environment where you can make sure that there aren't other variables, other factors, other things going on that are driving the changes that we're seeing. So when we had talked about that third variable problem in correlational research and how we're not sure if there's something else going on, experiments are going to hold all other potential third variables constant so that we know that they're not involved and that they're not confusing our results. We've already talked about a variable being a property or a trait whose value can change across individuals and over time, but we're going to go a little bit further when talking about experiments and talk about independent and dependent variables. So our independent variable, sometimes shortened to IV, is the variable that we're manipulating in an experiment. So if you have groups of people and some people are shown a TV show and other people are made to listen to the radio instead, what type of media they're exposed to is the independent variable because we as the experimenters are controlling whether they're exposed to TV or radio. Our dependent variable, or DV, is the variable that we measure in the study, the thing that we're looking to see if it changes based on the variable that we've manipulated. So our dependent variable will change depending on the manipulation of our independent variable. Now, experiments can get fairly complicated, but we're going to talk about sort of the most basic types of experimental setups. And in this most basic type, you tend to have two groups, an experimental group and a control group. So we could have a group, an experimental group, that's going to receive treatment. Maybe they're exposed to radio or TV. Those would both be considered forms of treatment. A control group is not exposed to any kind of treatment. And our control group is basically there to give us a baseline. It's going to give us something to compare to. So a control group would be maybe there's a group that just sits in a quiet room. They don't have TV or radio, so they are not exposed to any kind of treatment. And we can see if the people who are exposed to either radio or TV differ from the people that weren't exposed to anything on whatever trait we're interested in measuring. Maybe on how bored they are or something like that. So you can set it up however you'd like. Now, to decide which of your subjects, which of the participants in your experiment end up in your experimental or control group, we have a special procedure that we would use to decide who goes in which group. And this group is this procedure is random assignment. And random assignment basically is a setup where each participant has an equal likelihood of being assigned to any one group within an experiment. So of the people who are going to be in your experiment, they have an equal chance of being in your experimental group or being in your control group. Now, this is going to start sounding kind of familiar. This setup seems very similar to what we had talked about earlier with random sampling. But for random sampling, that's how you're picking your sample by randomly choosing people from a population. So random sampling helps you get from a population to the sample that you're going to use, and then random assignment helps you get from the sample the people that you chose from the population, and random assignment allows you to assign them to be in either your experimental or control group. And we use random ways of dividing this up, so you could use a random number generator or something like that, flip a coin, pull names out of a hat, something random, just to make sure that we're splitting people evenly. Everyone has a chance to be in either group. You don't end up using maybe a biased method of assignment. I like to use the example of you're running an experiment, a psychological experiment. You're going to be doing a test to see if people who listen to Mozart perform better on a learning task than people who are forced to listen to metal music. And the first 5 people that show up for the experiment, they get to go to the computers that are set up for Mozart, and the last 5 people to show up, they get assigned to the computers that are for metal music. But what if the first 5 people that showed up share some other trait, some kind of third variable that might also cause differences between the groups? Maybe the first 5 people that showed up early because they have a personality trait that makes them keen to be early. Maybe they're studious students and wanted to be on time. What if that personality trait of being early makes them perform better on that particular test? If the one group showed up early and performs better on the test because of the trait that made them show up early, it doesn't actually tell you anything about listening to different kinds of music. So you run into that third variable-like problem with experiments as well. And so random assignment helps us mix up the people that come so that they don't tend to be grouped together by any particular trait. And again, for those who are visual, we can start with our sample of participants. And you remember this sample comes from our population, and we will randomly select our sample from the population. Then we use random assignment to assign our sample, the people in our sample, into either our experimental group or our control group. And this experimental versus control group is going to vary based on our independent variable, the thing that we are manipulating. So in this case, maybe they're going to have a bunch of people who are going to go into a VR simulation and drive a car around a track. And our experimental group will have to drive this virtual car while also reading messages that come up on a cell phone. Our control group will only have to drive the car. There's no cell phone exposure. So they are controlling for whether or not you have a cell phone. In this case, their dependent variable is going to be looking at braking time or reaction time. So how quickly are they able to respond to things that pop up in the road? So they're measuring if people in the group that had to use a cell phone are going to take longer to react than people in the group where they don't have a cell phone. And then we can statistically compare those two groups to see if there's a difference. And that's the basic setup for the experiment. Now, before we pat ourselves on the back for working through experiments, I do want to point out that there are 2 basic types of research designs that we want to talk about. So the first of these is called a between groups or between subjects design. And this between groups design is basically what we just talked about. Each group in the experiment is composed of different participants. So we have 5 or 10 people who were given a cell phone to watch, and they were set into the driving simulator and they did the task. There's another group, a control group, a different set of people who just did the driving simulation. They didn't have a cell phone. These 2 groups of people are different people. So that's our between groups. But there's another special case that we can use, and this is the repeated measures or within subjects design. And so from the title, repeated measures, basically telling us that we're going to measure something over and over again. And in this setup, each participant is going to be exposed to all of the conditions of all of your independent variables. So if we come back here, this would be saying that instead of having this group of sort of greenish people are our experimental group, and this separate group of orangish people are our control group, for a repeated measures design, we would have, say, 10 people, and the first thing that they would do is drive around the track with a cell phone. Then, after we got their measurements, we would tell them to do it again, but without the cell phone. So we would measure them a second time, a repeated measurement, and get the measurement for the same people without that experimental condition. So you would be looking at repeatedly measuring the same people. So your experimental group and the control group are the same people. They're just exposed to both conditions at different times. For a different visual representation, we would have our between groups. So group number 1 gets treatment number 1, driving with the cell phone. Group number 2 gets treatment number 2, no cell phone. Whereas with repeated measures, you have group number 1, and they're given treatment number 1, driving with the cell phone, and then treatment number 2, without the cell phone. Now, if you downloaded the slides for this early, I had these backwards, where I had these images down here and this image up there, but I have fixed it now, so if you care a whole bunch, you can re-download this to get them in the right order. My mistake. And also, very briefly, I want to touch on the fact that we've talked about having one independent and one dependent variable. You can also have experiments with multiple independent and dependent variables. So it gets way more complicated than what we've talked about, but we wanted an example. So maybe we wanted to look at the same idea, people who drive with and without a cell phone, but we also want to see if there's something going on with traffic density. So not only are we going to manipulate whether people are driving with or without a cell phone, we're also going to get them to drive in low density traffic or high density traffic. So the idea here is that, well, maybe there's something going on where people are better at driving when there's low traffic density, and they're worse at driving in high traffic density. We'd also think that they're probably better driving without a cell phone and worse driving with a cell phone, but by having these two independent variables, if you set up your experiment just right, we get to look at something on top of just the two things we wanted to look at. And that extra thing is the interaction that's going on. Maybe there's something more than just those two variables. And if we look at the data over here, where if we're looking at this bar graph, we have traffic density. So these two bars here are for the low traffic density, people who drove when it wasn't very busy on the roads. And these two are high traffic density. We also have their braking reaction time, so how quickly do they respond to something in the road, where a lower score is going to be better. And then we have our green and purple, so just driving, and purple is the cell phone. So lots of stuff going on here. But what we care about is that in low traffic density, people with and without a cell phone have about the same skill level. So you're not overly impeded by driving with a cell phone when there isn't anything on the road. Kind of makes sense. But when there's high traffic density, when there's more things on the road, people who are driving with a cell phone take much longer to respond. And so the fact that there's no difference between the two groups here, but there is a difference in this condition, tells us that there's an interaction between our two different independent variables. So you can extrapolate this and start looking at 2, 3, 4, 100 independent variables. It just gets more and more complicated as you get more and more of them. Now, we've talked a lot about finding differences between groups or finding correlations between two different variables, but we haven't really talked about the statistics side of how do we know that there's a difference? When do we say that there's a difference between the groups? I can actually go back here and say, well, this green bar is actually smaller than this purple bar. Why would you say that there's no difference between these two groups? And why is this big enough to be a difference? How do we decide how large of a gap is a big enough difference? And that brings us to statistics. And specifically, we're going to look at inferential statistics, where we're going to test the significance of differences between those groups. So we want to see if the effect that we're observing, the difference between those two groups, is meaningful, if it's significant. And so we can talk about significance as the likelihood that a relationship between two or more variables is caused by something other than chance, which is basically a fancy way of saying we want to look at the odds that the difference between these two bars is caused by the things that we're manipulating. That it's not just some fluke, some random accident or random happenstance that led to the difference between these two bars. So significance is telling us how sure we are that this difference is because of the manipulations that we did in the experiment and not due to chance. Some sources will say, instead of due to something other than chance, they'll say that it's happened because our random assignment failed. Basically saying that our groups were different on some other factor, because random assignment didn't give us equal groups on every factor. But they're both saying about the same thing, which is how sure are we that what we're seeing is caused by the manipulations that we're doing? And a good way to think about this is a simple probability question. So think of a coin toss. Random, it should be random if you don't have a weighted coin, but if you flip a coin, 50% of the time you should get heads, and 50% you should get tails. But when we talk about probability like that, that's across infinite trials. We're saying if we flipped a coin forever, half the time it would be heads and half it would be tails. But what about in practice? If we flipped a coin 100 times, and we observed any of these three options, a 70-30 split, a 50-50 split, or a 30-70 split. For the 50-50 split, we'd probably say that, yeah, that's exactly what we expected. There's nothing else going on. If we see these 70-30 or 30-70 splits, is that significantly different from what we expected? Is that different from 50-50? So that's where our inferential statistics come in. And we're not going to go into calculating statistics for this because, again, there are stats courses for that, and it gets very complicated very quickly. But I just want to expose you to the idea here. And if we decide that this 70-30 split is significantly different from our 50-50 split, what are the rules that we're using to determine that? So our convention, the accepted standard, is that you can't claim to have significant results, that you can't claim that your results are meaningful, unless you can report that you are 95% sure that random assignment has not failed, or you are 95% sure that what you're observing has happened because of the manipulations that you made. And if you're 95% sure, that leaves us with 5% of uncertainty. And that's where we get this P of 0.05. So our statistics are usually going to report a p-value. And the smaller that value, the more sure we are that the results that we observed were due to our manipulations, and they weren't due to chance. They weren't due to failure of our random assignment. So P of 0.05 is our cutoff. Smaller than that means that we can say that we have a significant difference between our groups, that we are significantly sure in what we are observing.